Hello, and thank you everybody for joining us today. My name is Sven Bocklandt, and I'm Senior Director of Scientific Affairs and Marketing here at Bionano Genomics. And I would like to welcome you to this webinar brought to you by LabRoots and sponsored by Bionano Genomics. The webinar is called Isolating Ultra-High Molecular Weight DNA for Genome-wide Structural Variant Analysis, and it will be presented by Dr. Ben Clifford and by me, Sven Bocklandt. Bionano has been working on the isolation of megabase-sized DNA molecules for over a decade for our optical mapping or genome imaging technology, and we were hoping to share some of that experience with you today. First up is Dr. Ben Clifford, who is our Senior Field Application Scientist. He will talk about all the tips and tricks for sample handling and DNA isolation. Then I will take a few minutes to show you the things that you can do with that ultra-high molecular weight DNA that you just can't do with shorter molecules. And finally, we will answer as many questions as we can. We encourage you to submit as many questions as you want at any time you want during the presentation. To do so, just type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. With that, Dr. Clifford, the virtual floor is yours.
Thank you, Dr. Bocklandt. Hi, everybody. Thank you so much for joining. My name is Ben Clifford. I'm Senior Field Application Scientist at Bionano Genomics. And for anybody who is not familiar with Bionano, our platform is pictured here on the title slide. We have the Saphyr system. As Sven said, it does whole genome imaging. So it's not a sequencing-based approach, it's genome imaging. And the reason that that's important is that genomes are highly repetitive. And so if you look at just a breakout of the contents here of the human genome, only around 1/3 of it consists of unique sequences. The other 2/3 are repetitive elements, so lines, signs, transposon, anything else bound in repeats. And so shorter technologies like sequencing can often get confounded by these repetitive elements and lose context for what an individual read might be covering. And when we talk about structural variation, we're talking about the largest events in the genome, chromosomal rearrangements or large, oftentimes, events. So here, insertions, deletions, out to inversions and even translocations at the chromosomal level. And because of the challenges with NGS, for example, the clinical standard of care for some of these largest events has been the karyotype. But that's slow and not particularly scalable. And so really where we sit is bringing some of the same advantages that sequencing data applies, the scalability and the event detection precision. And that's where Bionano's data type is particularly valuable. So here's an image from the Saphyr system. This is showing high molecular weight, long intact DNA molecules in our nanochannel arrays that have been labeled and stained with our labeling chemistry. These fill in for reads, but they are much, much longer. And this approach really requires the long molecules. In fact, we only carry forward molecules longer than 150,000 base pairs in equivalent, 150 kp. And this is really what allows Bionano to assemble entire chromosome arms, span these long, repetitive, complex regions that are often confounded by shorter approaches. In fact, in the image here on the right of the slide is some more of our data, and you can even see, visually pick out some repeat motifs. So there's a few strings of labels that are consecutively shown, and we can span most of those regions as well as flanking regions to identify where those repeats exist in the genome. This approach really requires Bionano Prep DNA isolation to attain the long, ultra-clean DNA. I'm sorry to say, if you have a freezer full of DNA that's been archived, that's been isolated from other methods, then frozen, it's just going to be too short. Short molecules and impure samples just aren't likely to be informative once they're labeled and imaged in this way. Either they're too short and that native information from the length is lost, or the prep is too dirty and it won't label efficiently like we need it to and be informative for the downstream assembly process. And so the rest of my section is really going to be addressing sample selection, selected your samples, and gently handling the sample. At the sample and DNA level, those factors are critically important when it comes to prepping this DNA. And what you get in following these best practices, here's a histogram of molecule lengths coming off of the Saphyr instrument. So this is a particularly good run. This has a molecule N50, so a center of mass of the size distribution, 434 kb, 434,000 base pairs. If you look to the left, this red circled area, this is typically what you get with long-read sequencing approaches, 20, 30 kb, sometimes a little higher, and 50 lengths. Our center of mass is typically much, much higher than that. Really anywhere 250 kb and up is typical. We will often speak in terms of N50 for where the center of mass of distribution is, although some molecules in the distribution go up past megabase, even past 2 megabase, 2 million base pairs in length. But what's really informative for our downstream assembly process is where the center of your data set resides in terms of length. And then just to take a sample that's processed through the Bionano Prep, if we look at that on a pulse field gel, that really gets at the size, even before it hits the labeling chemistry or the instrument. So here's a pulse field gel. These bottom two rows are native DNA that have been isolated via Bionano Prep methods. And the important thing to look at, this rather long ladder goes to 825 kb. If you look at the banding pattern on the bottom two rows before that, there's DNA present in what we call the megabase compression zone. So these isolations contain megabase DNA. The top two rows here are the DLE1 labeled DNA samples that are going on the chip. They also have megabase DNA at the compression zone. That's megabases that you're loading into the chip that can then potentially be imaged on the Saphyr. And so detecting structural variation really starts with these long DNA molecules. And we have a number of protocols now using solution phase prep. Our previous methodology for this was based in plug lysis, which I just want to address briefly. This is using an Agarose Gel Plug to provide megabase-sized DNA and physically shield the sample. So what that starts out as is cellular material with its native state DNA in it. And then you're providing an agarose gel matrix of protection around it. And from there, there are a series of lysis steps where the cellular material, anything that's not DNA, can be lysed and digested away with proteinase K and lysis buffer. And there, after a series of wash steps, the agarose itself can be melted away. And then you're just left with high molecular weight DNA solubilized. This is not very scalable, however, or very fast. It's a number of hours and an overnight incubation, and we wanted to be a bit faster than that. But the problem with most fast DNA protocols, whether you're using a spin column or magnetic beads or precipitating to the bottom of the tube, is it's just too much mechanical force on this native fragile DNA, and it gets you short reads. So we developed a Bionano Prep SP solution phase DNA isolation protocol that's both fast and mechanically protective of the DNA. And just to outline how it works, we take a biological material from one tube and then undergo a lysis process and gently release DNA into solution. It's then bound on a magnetic disc where it can be washed on the disc and then transferred to a new clean tube once it is clean and then eluted in an elution buffer. And just to give you a real example of what this looks like, here are a couple of tubes with two black magnetic discs in them, and then each one looks like it's bound up in cobwebs a bit. So this is what the DNA looks like precipitated out of the solution and then bound magnetically to the disc. You have these white wisps that just attach to the disc. It can all be washed as one unit and then transferred to a new clean tube. And so this process really, it's critically important to handle the sample carefully, store and treat the sample carefully. We do tend to see our very longest molecules with samples that are fresh and have never been frozen, but there are freeze options as well for the following sample types: whole blood, bone marrow aspirate, cultured cells, and then new to the lineup, tissue. So we have a solution phase approach in human and animal tissues, tumors, using SP. And then we do have workflows for plants as well. Those are currently supported with Agarose plug based workflows. I'll go through each of these in turn. So for whole human blood samples, we recommend these be drawn into EDA tubes. And then there is an option to process that blood same day, fresh, without freezing. And what that entails, if you are going to store it for any length of time, that it be stored at 4 degrees Celsius and then prepped within 4 days of that initial draw. If that's not possible, or if you'd rather store blood and get to it later, there is a freeze option with this workflow. That would be to mix the original stabilization tube and then make small aliquots. 650 microliters is really all it takes. And then place those at -80 and have those available when you're ready to work with it. This workflow is supported by, shown below, the Bionano SP Prep Blood and Cell Culture Isolation Kit. There's a similar workflow available for bone marrow aspirate. Now, bone marrow aspirate's a bit challenging. It can be a complex sample type just due to the nature of sample collection. So we've added a filtration step. So occasionally there is bone shard, sometimes skin or fat left over in the sample. So we want to remove all of the crude bulk material and just leave the white blood cells. Another possible complication with bone marrow aspirate is these are collected typically in heparin tubes. Heparin, for our purposes as a stabilizer, just is not very effective in stabilizing the cells through a freeze-thaw cycle. So what we recommend is to add in a reagent DNA stabilizer, 15 microliters per 1 ml of crude bone marrow aspirate. Titrate accordingly if your volume is slightly different. We see our best results and strongly recommend adding this while the bone marrow aspirate is still fresh before it's frozen, and it can be mixed together well. If bone marrow aspirate has been frozen beforehand, quickly upon thawing, you can dose in an aliquot of DNA stabilizer. That will add some benefit. But typically the easiest way to work with this material is to mix it up in DNA stabilizer and make 1 ml aliquots, which then can be placed at -80. This workflow is supported by the Bionano Prep SP Bone Marrow Aspirate DNA Isolation Kit, which includes those filters and the extra DNA stabilizer. Cultured cells are also quite readily handled. If you're electing to work with these fresh cells in culture, the culture should be high viability. Ideally, it's actively growing, in which case you would simply count out 1.5 million human cells and begin the protocol. So you'd carry the volume equivalent of 1.5 million forward after doing a cell count. This 1.5 million figure, this is to target 9 micrograms of DNA equivalent going into the reaction. More on that in just a second. There's also a freeze option with cultured cells. If you have cells in culture and you'd rather freeze them down into pellets, that's certainly possible. You'd simply count out your desired input and then wash again in this DNA stabilizer to promote viability in those cells. Upon freezing them and thawing them back out, you'd pellet the material once it's been washed in stabilizer and then place those tubes at -80. And this workflow is supported by the same kit as the whole blood, the Bionano Prep SP blood and cell culture isolation kit. Now, each of these 3 workflows that I've described, blood, bone marrow, and cell, have a number of commonalities that I want to address just to get into the mechanism a bit more specifically. In each one, the cells at some point are counted to 1.5 million human cells as the target input. This translates to an input of 9 micrograms of DNA equivalent of the material. We like 9 micrograms as a target, as that provides enough material to have a good DNA yield, a target concentration of the solubilized sample once you have it as output. But it's not an excessive amount of material. It's less than 650 microliters whole blood, for example, and really minimizing unnecessary input is helpful for the efficiency in the wash steps. So you're minimizing how much there is to wash out. In every case, cell viability is really key. If the cells are inviable in a significant degree, if nucleases are already attacking that DNA, you're going to see shorter molecules once you get to the labeling and running the Saphyr. And so for electing the freeze options here, that's where stabilizing the sample properly really comes in. And then once any frozen sample is frozen at -80, samples really must not undergo any freeze-thaw cycling. So those are freeze-thaw cycles just represent iterations of damage to the DNA. Now, I mentioned at the outset, we have early access kits and workflows available for human and animal tissue. Just to go into that briefly, we were seeing good results with 10 milligram and up of flash-frozen tissue or tumor. Unfortunately, FFPE samples are not adequately preserved and won't work on our platform. But once the desired mass of tissue is harvested, it's simply a matter of rinsing it and quickly snap freezing in liquid nitrogen upon harvest. You don't need a storage medium. We don't recommend using any kind of storage medium outside of just snap freeze and liquid nitrogen. Let that gas off, cap the tube, handle it on dry ice until the sample can be transferred to -80. This workflow is, again, in early access. It's supported by the Bionano SP Prep Tumor and Tissue DNA Isolation Kit. If you are interested in this workflow or in any of our workflows, please contact us. And then we have a number of workflows available for plant tissue as well. So each of these start with harvesting anywhere between 0.5 and maybe 2 grams of fresh growth from young plant leaf tissue as input. With plants, each one can be very different, but one commonality is you really want to target young, fresh plant leaf tissue. Mature plants, leaves that have grown a lot of structural compounds or metabolites, secondary metabolites in the tissue, are likely to lead to dirtier preps, lower DNA yields. The fresher you can get, the better with these. We have a number of workflow options to support different plant compositions and metabolite content. Our workhorse protocol is a protocol that works well for most straightforward plant tissues. There are separate workflows specific to secondary metabolite content, a high polyphenol workflow, as well as high polysaccharide, if those are issues to be concerned about with your particular plant sample. As with the other workflows, fresh works great, in which case you just harvest the tissue and then start straight away, homogenizing fresh plant tissue. If you're not ready to get to that same day, there's freeze options here as well. You'd harvest the tissue, pre-weigh it out, and then snap freeze in liquid nitrogen. Again, no storage media really necessary to do this. Just snap freeze in liquid nitrogen, let that gas off, and then transfer a sample to -80 until you're ready for it. If the sample is frozen at this point, there's also a workflow option to homogenize the sample entirely in liquid nitrogen. This is just pulverizing it down into a powder and then using a weighed powder to carry forward into the DNA isolation reaction. These workflows are all supported by the Bionano Prep Plant DNA Isolation Kit. Again, this is an agarose-based method, plug lysis, and there's a protocol selection guide if you're not sure where your plant falls that will help you differentiate between whether it's better to use base, high polyphenol, or the high polysaccharide option. So the output from any of these processes is solubilized ultra-high molecular weight DNA. And then working with this and preserving that native length that you've worked so hard to attain entails a few extra care steps as well. So if you look at the picture on the right, this is what a high molecular weight DNA sample looks like. The first thing you'll notice is the texture is very different. The sample is viscous, so it's a bit even just like pipetting hair gel. There should be some texture and viscosity to it. Any of the SP workflows end the isolation protocol on a slow end-over-end DNA rotation to gently promote some mechanical mixing, and then that sample is incubated overnight, at least overnight, at room temperature. Returning to it the next day or a couple days down the line. It's critical to pipette slowly and very carefully, and really to work with the viscosity. So realize it's not going to be a uniform subsample when you pipette it. Just need to work with it. You never want to vortex the sample. That will shear your DNA systematically, and then it's strongly recommended to keep wide orifice tips on hand. So in this picture, technicians using a wide bore P200 tip. The orifice in the tip is wider, and that's to minimize any shearing of just the sample migrating in and out of the pipette tip. That's what we recommend for mixing prior to any subsampling for DNA quantitation or labeling this DNA. And then the gDNA that you do recover from this process should be kept fresh. You should never freeze it. The ice crystals will shear the DNA. Storage at room temperature is just fine. If you're going longer term, storing the sample at 4C also works. One other thing to keep in mind with these viscous DNA samples is that the DNA within them is often heterogeneously distributed. So the viscosity in the sample really is molecular interaction of high molecular weight DNA, long strands interfering with one another and providing a viscous texture to that sample. And that implies, and is often the case, that the sample is really quite heterogeneously distributed. When it comes time to quantitate that DNA, what we do is 3 sample aliquots per tube, small aliquots, 2 microliters, to quantitate using fluorimetry. And we do need a fluorimetric approach to get this quantitation accurately. And then from those 3, assessing the mean and the coefficient of variation. And just to illustrate some of the challenges in doing this, here's an example of 3 different aliquots of DNA from the same tube. So spatially different positions. You can take from the left, center, and right of the sample, just to ensure you're not hitting the same spot 3 straight times. 3 different readings from a same tube might look like 65, 100, and 120 nanograms per microliter. That's real variation within that sample. If those were your 3 readings, what you'd do is take the mean concentration of those 3, here it's 95 nanograms per microliter, and then of those 3, assess the coefficient of variation, so the standard deviation divided by the mean. We require a coefficient of variation 30% or below to have confidence in the initial 3 aliquots that went into this reading. So they need to agree within 30% or so of one another before having confidence in the mean. If the DNA is incubated overnight and then 3 aliquots are subsampled and they are wider than 30% in coefficient of variation, that's not a static metric by any means. The cure for that is simply just to let the DNA incubate longer at room temperature. What happens then is it gives the DNA time to diffuse apart and homogenize the sample. Some gentle mixing there with a wide bore tip is also an option. But just to underscore where these processes leave you, you end up with a very high ultra-pure molecular weight DNA isolation. The initial input target was 9 micrograms DNA equivalent. And that's typically output in about 120 microliters of volume, which represents solubilized ultra-high quality DNA that you can carry forward for labeling. The typical concentration, the target that we aim for with that 9 microgram input, is 35 to 150 nanograms per microliter in mean concentration. Once you're at that point, if you're doing the Bionano workflow, it only requires 750 nanograms of DNA carried forward to be used in the direct label and stain reaction for running in the chip. So we're producing quite an excess of DNA that is actually run in the instrument. And with that, I'd like to hand things back to Sven to talk a bit about the platform and structural variation in genetic disease and cancer. Sven.
Yes, thank you, Ben. I would like to start by summarizing a few things that Ben said, and that is really that one of the reasons why we are all here and are interested in these very long DNA molecules is that the genomes of any complex organism are just really highly repetitive. In human genomes, only about 1/3 consist of unique sequences and 2/3 consist of repeats. And what's important here is that if you have a short read length, because you have short molecules, but also because you're using a method like NGS to look at the genome, you just cannot make sense of 2/3 of the genome because almost all of the classes, all of the single units of these different repeat element classes are already longer than the length of short read sequencing. So making sense of that is impossible. And even with long read sequencing, you're still not able to make it through some of these long repeat arrays here. And that's why the standard of care in the clinic is not long read sequencing. It's not high coverage Illumina, but to look at structural variants. People look with a microscope at intact DNA molecules. And when NGS came along, there was this idea that all of this could be replaced with short read sequencing, but the truth is that an NGS protocol starts by fragmenting the DNA. You take a perfectly intact genome and you shred it. And that makes no sense. It's the same idea of trying to tell you how many pieces of kiwi were in this blender after I turn it on and make a smoothie. It just doesn't make sense. So we want long intact DNA, and that is what this seminar, of course, is all about. That's why the standard of care in the clinic is still looking at intact DNA molecules and looking at patterns on these molecules, and that's exactly what Bionano does. We place about 1,000 times more labels on these molecules than you can see with a karyotype, and we massively parallel image and linearize these DNA molecules in our nanochannels array. Our microscope is called Saphyr, and it automates the labeling, the imaging of these molecules. Now, this is how it all works. This is the workflow for optical mapping with Bionano. Ben talked extensively about the isolation part of high molecular weight DNA. Next up then is labeling a specific sequence motif across the entire genome. We do this with a single enzymatic reaction that places a label at a single 6 base pair sequence motif occurring all throughout the genome about 500,000 times, so once every 6 kb on average. Once that labeled DNA is loaded onto our Saphyr chips, we will use electrophoresis to move the molecules into the nanochannels and image them, and do this over and over and over again. The images that are collected will then be digitized, and the digital representation of each of the molecules will be extracted. And then our software will perform a complete de novo assembly of the entire genome, meaning that every single molecule that we imaged will be aligned with every single molecule that we imaged. And so out of these patterns that we find in these overlapping molecules, we build consensus genome maps that represent the structure of the genome. And then we can call every type of structural variant simply by comparing these patterns. In each of the examples here, the reference genome of your choice is in green and your sample is in blue. And you see on the left that a gain or a loss, so a deletion or an insertion, is detected by the spacing between the labels, decreasing for a deletion or increasing for an insertion. With Bionano, we can call those starting at 500 base pairs. We can look at repeat arrays. I will show you some examples of that in a second, either by counting the repeats or by measuring the distance between the flanking labels. We can detect tandem duplications by seeing a pattern present twice next to each other, either direct or inverted, starting at 30 kb. On the top right there, you see a translocation, which we detect when the de novo map in blue aligns for half with one chromosome and half with another chromosome, or a different part of the same chromosome. And then last, bottom right, an inversion is detected when a pattern is simply flipped around in place, starting at 30 kb. Our software will show you all the structural variants that we detect in a figure like this, which is called a circus plot. You see translocations in the middle, magenta, and then the concentric circles show you copy numbers, deletions, inversions, duplications, etc. Now, if you click on any of these structural variants, we will show you a zoomed-in map that shows you exactly the alignment of that structural variant with a reference. And you can even show the molecules that were used to call that structural variant. Now, if you don't want to use with our software, you can simply export these structural variants as a VCF file after filtering and use any software of your choice. Now, what makes Bionano unique compared to long-read sequencing technology is really the enormous amount of data that we can collect in a short amount of time. Our chips have 3 flow cells, so you can map 3 genomes at a time. And the instrument can collect the 100x coverage, which we recommend for structural variation calling in genetic disease, on 9 human genomes per day per instrument. Or if you want to collect more data, you can run them longer and collect as much as 400x coverage, which we recommend for cancer. And I'll tell you in a minute what we do with that. We can run the instrument even longer and collect 1600x coverage of 3 human genomes in just 3 days on average. An enormous amount of data. And keep in mind that the cost per genome, if you have an instrument, is just about $450 for that. So 1600x, $450. I think if you work with long-read sequencing, you immediately understand that there's an enormous gap there in what we can deliver compared to other methods. So I want to walk you through a few examples of structural variants that were detected with Bionano that were missed with other methods, short-read and long-read sequencing. And I will start first with some genetic disease research cases. What you're looking at here is a part of chromosome 16 in a patient with congenital diaphragmatic hernia. That's a severe genetic disorder, a birth defect that affects the diaphragm and the lungs, sometimes the heart. And very little is known really about the genetic basis of that. So at Mass General Hospital, a team used Bionano to look at this case where a microarray had detected a 2 megabase duplication. Now, if you look on top of that figure, you see a copy number profile, and our copy number profile shows actually 2 duplication events. And then when we look at our maps, the patient map is in blue, the reference is in green here. You can see that we can show you the exact structure of this event. There's actually 2 duplications. The first one is direct, the other one is inverted and falls in between the first 2 duplications. We highlighted the genes that are affected by these events, and those are all new candidate genes for this disease. They could not make sense of this region with sequencing Mark Ebert from the Mayo Clinic used our technology to look at the C9orf72 gene, which can cause Alzheimer's disease if there's an expansion of a GG-GG-CC repeat. That hexanucleotide repeat is, of course, all GC, 100% GC, and so these expansions of that repeat are impossible to sequence through. Mark tried to do this with nanopore sequencing and, of course, short-read sequencing as well, and failed. Here you see that in a single brain biopsy of a patient who died of ALS, we can see these different alleles, these different expansions of that repeat from wild type all the way to 32 kb. And there's, of course, an enormous amount of power in being able to detect that mosaicism in the brain. Here's another one that you cannot sequence with short or long read sequencing. It's a disease called FSHD, facioscapulohumeral muscular dystrophy. It's a genetic disorder that affects the muscles of the face, the upper arms, and the back. And it has a very odd genetic mechanism. It is caused by repeat contraction, a contraction of a repeat subtle American chromosome 4, which is labeled here in this figure as D4Z4. You see a wild type on top, and you see a diseased allele at the bottom where you see contraction of that repeat. This repeat is in a healthy person up to 300 kb in length. Bionano can span that repeat with single molecules. That is really our strength, our megabase size molecules. And so we can size that perfectly in single molecules, and we can also distinguish between the pathogenic and the non-pathogenic allele, which is labeled 4QA and 4QB here. Segmental duplications, or low copy repeat LCRs, as they're labeled in this figure, are very, very large blocks of the genome that are repeated multiple times. They're typically 50 to hundreds of kbs in length, and they can be blocked together in blocks as large as a megabase or so. Bionano can span these segmental duplications with single molecules, and so a team at Emory University used our technology to look at inversions flanked by segmental duplications. In the 3q29 deletion syndrome, a large 1.5 megabase section that is labeled in this figure as canonical deletion is deleted and causes the disease. But this team wanted to find out if inversions in the parents in that region can predispose to this disease. They tried to do this with just about every molecular method that exists, but only with Bionano they were able to detect these inversions because we have molecules that span the 350 kb inversion and both segmental duplications that flank it on single molecules. Mark Ebert, again at the Mayo Clinic, has talked in several papers about camouflaged genes. These are genes that have often repeating exons that make them impossible to be analyzed by NGS. At the bottom there in orange, you see the read depth from whole genome sequencing or exome sequencing for this gene, and you see that it drops to 0 for the binding domains of this gene, again, because there are variably repeated binding domains in that gene. This gene is very important for Alzheimer's disease, and with Bionano on top there, we were able to call a deletion in this gene in a patient who died of Alzheimer's disease, even though the repeats there make it impossible to access them with sequencing. Now in cancer, whether you're studying heme malignancies or solid tumors, we really have a great advantage over these other technologies, and that is, I mentioned before, that we can collect as much as 1600x coverage per sample for the same price. With that, we can dig very deep inside a complex heterogeneous tumor and detect structural variants that are present at just 1% allele fraction. No other technology can do this in an unbiased genome-wide manner. And so 2 examples from cancer samples. Here is a uveal melanoma sample that is characterized by typically a loss of expression of gene 1. This is a result from the Curie Institute in France. In this particular sample, they found no genomic explanation for this lack of expression, but Bionano was able to detect a deletion of 740 base pairs right in the promoter region of this gene. Now we plotted the GC content there underneath, and you see that right at the site of that deletion, the GC content goes up to about 80%, and that made it invisible to the methods of whole genome sequence that they used. Here's another example from the Cordelier Institute, where they studied hepatocellular carcinomas. On the left there, you see the circus plot of a tumor that showed no replication stress. You see a few translocations and a very stable copy number profile. And on the right, you see a sample that looked identical under the microscope, but had an enormous amount of replication stress. You see a massive number of translocations and also a highly abnormal copy number profile. Now, the difference between these two was the insertion of the hepatitis B virus just upstream of the cyclin E1 gene, which you see in the map on top there. We call this insertion, and so the promoter of the hepatitis virus will turn on that cyclin gene. The cell cycle will continue out of control before the DNA replication has happened, and that causes that signature. This is an important measure, this signature of replication stress, because it may qualify this patient for the use of PARP inhibitors. And then last, I want to touch for just a second on cytogenomics, because in cytogenetics right now, we use decades or even half a century old technologies, karyotype, FISH, chromosomal microarray. And a recent study that was just published from Radboud University in Nijmegen used Bionano to look at 48 leukemia genomes and found that Bionano can call all the variants, so 100% concordance with the standard of care. That means all the variants picked up by karyotype and by FISH and by array in all 48 samples combined. Many sequencing-based methods have tried and failed to replace traditional cytogenetics, but Bionano can. And the advantage here is that you can replace a very confusing, to be honest, workflow that you see on the left, where depending on the tissue type, you end up with cell culture or not, cell separation or not, different methods of DNA extraction and of labeling and of amplification and of harvesting and hybridization to come up with a number of different results that then need to be integrated. All of this can be replaced with a single workflow where you have a universal DNA extraction independent of the sample type, as Ben discussed with our SP kits, and then one instrument generates the data that gets analyzed all together. So I want to end here by summarizing that we call structural variants starting at 500 base pairs genome-wide with extremely high sensitivities up to 99% and PPVs starting at 97%, so almost no false positives. And then in cancer, we can collect 1600x coverage per sample. That brings us down to 1% allele fraction. All of this just starting at $450 if you own an instrument. But that's not the only way to get Bionano data. You can, of course, get the Saphyr system shown on the right there. You can also take part in a reagent rental model where, with the commitment of just 120 genomes at $550, we will get you a Saphyr instrument to use. Or we also have an in-house service where you simply send us your samples and we will get you the data back. And we are up and running even right now during these times. So with that, I want to thank you for your time and address some of the questions that we've received. And I will start to ask Ben a question about size selection. Is there any size selection mechanism of the disk at all, Ben?
Thanks, everybody. Thanks, Sven. Is there any size selection of the disk? No. The disk is introduced once the input material has been lysed, and then megabases of DNA diffuse into the solution. The disk simply captures what's there. So it's not meaningfully performing any size selection.
Another question, Ben, will the magnet affect the DNA in any way?
So the DNA binds to the magnetic disk. It's not a pull in opposite directions like you can see with bead-based approaches. It's just a magnetic disk with a microstructure that enables the DNA to bind to it out of solution. So it's not exhibiting the mechanical force of other approaches.
I'll stay on the disk here. The magnetic disk that allows binding of DNA resembles Circulomics nanobind disk. Is that the same composition, or what's the difference in the protocols?
That's right. So it is the same composition, but they are different protocols. So we have partnered with Circulomics and developed workflows specific to our needs of DNA purity, and developed the workflows and specific chemistry around the Bionano Prep SP DNA isolation.
Great. We have a few questions about yield. The extraction with 650 microliters of blood will yield a very low final yield. It may be long reads, but not enough DNA for multiple reads. What is the typical yield of 650 microliters of blood?
That's right. So I'll take that one step by step. So 650 microliters of blood. A small sampling of that is used to determine a white blood cell count. And then from there, the volume equivalent of 1.5 million white blood cells is carried forward. So that's really a commonality between cell, blood, bone marrow aspirate. We use 650 microliters because that's a pretty conservative high volume of blood. Whole human blood from healthy normal patients, anywhere from 250 microliters to maybe up to 500 microliters will get you that 1.5 million. Now, if there's any factor where the white blood cell count would be abnormally low, you could simply potentially titrate a different volume just so long as you can isolate 1.5 million white blood cells. And then once you started at that common point, you proceed through the isolation, and then the target output is anywhere from 35 to 150 nanograms per microliter in concentration.
Great. Somewhat related question. A scientist here stores frozen blood samples in 1 ml volumes. Can we scale up from the recommended 650 microliters?
Potentially, sure. So in that case, if you wanted to follow the workflow to a T, you would simply take that 1 ml subsample and from it see what volume equivalent gets you 1.5 million white blood cells. And then you'd simply have an excess of blood that you could use toward whatever other purpose.
Yeah. How do you quantitate this kind of DNA? What method is used?
Great question. So quantitation, we just need some fluorometric approach. So in the field, typically we'll use the qubit fluorimetry. That picogreen works as well. An approach like nanodrop isn't going to be precise enough, unfortunately.
Yeah. Okay, we have another question here. Is there any difference between high molecular weight DNA and what we are calling ultra-high molecular weight DNA? For human blood samples, do the separation techniques differ between these above-mentioned 2 molecular weights DNA?
No. No, so there's not meant to be a difference in terminology there. DNA is, we want it as high molecular weight as we can get it.
I think what I can add here, maybe, Ben, is that many DNA isolation kits claim that they isolate high molecular weight DNA, and you get molecules that are maybe 30 to 50 kb in length. And I think the difference is that at Bionano, we require or want megabase size molecules. That's where that distinction falls, I believe. Does the DNA isolation protocol work for single-stranded DNA?
No, so the output is around a target of 9 micrograms double-stranded DNA.
Yeah. Similar questions for viruses. Does the protocol work for DNA isolation from viruses?
So viral genomes are going to be much smaller than the target molecule length for Bionano Prep. So we haven't explored whether it could work in principle, but you know, much of the benefit would likely be lost.
Okay. We have quite a few questions here, so I will just keep going, if that's okay with you, Ben. The grinding of plant tissues isn't affecting DNA fragment length at all. Methods like chopping the tissue may be better. Is the grinding affecting fragment length?
Yeah, let me put up my plant slide again. That's a very good question. So I mentioned a few separate workflows that we have for plant tissue. If it's processed fresh and kept that way, each of the three shown here, the base, the high polyphenol, and the high polysaccharide, those use chopping to homogenize the plant tissue, and then that's processed through a plant homogenization buffer, which releases the nuclei. That is certainly one option. Liquid nitrogen grinding is a separate one. I'd recommend it if the plant tissue were stored frozen. If it's already been frozen, you may as well keep it that way in liquid nitrogen for the duration of homogenizing it, in which case it's easiest to grind. We see that it's more important really to keep a lot of liquid nitrogen around and to keep refilling and keep that tissue cold during the grinding process. If it's spiking in temperature as you're grinding it, that will have an impact. But if the liquid nitrogen, if there's plenty of it and you're able to grind it away, it's sometimes necessary if the plant tissue is particularly tough, or otherwise. But really, there's an option if there's a concern about treating the tissue too rough. You could certainly elect to chop it. But in the realities of plant tissue is it's toughly used sometimes, and the grinding can be a benefit just to get through it.
Great. Another question here. What is the maximum time after tissue snap freezing for high quality genomic DNA to be able to be recovered?
That's a good question. We, to my knowledge, haven't looked at this systematically. I think the ultimately important thing is that it be snap frozen and then kept at -80 without freeze-thaw cycles. Really, at that point, I would imagine it comes down to particularities of that sample and of your freezer maintaining that temperature.
Somewhat related, how should I keep high molecular weight genomic DNA after extraction from a cell? In 4 degrees or at room temperature, how long can I keep it in ideal conditions?
Sure. So great points both, great options both to keep it at room temperature or 4C. The important thing is you don't want to freeze it once the DNA is solubilized. Just as a workflow convenience, typically our users are turning that sample over into labeled DNA and then running it out on a chip pretty promptly. If you do want to store it, room temperature is fine, maybe out for a couple weeks. Anything longer term than that, you can put it at 4C, but it's pretty user electable in practice.
Great. We have a few more questions about the SV calling that I can take. What's the repeat expansion detection sensitivity? Now, it's an interesting question because we can automatically call structural variants starting at 500 base pairs. We cannot see very, very small expansions or contractions of repeats. So if you just have a few copies of a trinucleotide repeat, we cannot detect that. So the type of repeat expansion genes that we can detect are the ones that are larger and that typically fail with PCR. So there's some good complementarity there. That is Fragile X, myotonic dystrophy, ALS, and a number of other ones. So if the difference between the wild type allele and the expanded allele is more than several hundred base pairs, we will be able to detect that. Another question here, it says the starting size of SV detection is 50 kb, but what is the range of SV sizes? So a little correction there, we call structural variants starting at 500 base pairs. It's on the screen right now, actually, for deletions and insertions. For some of the other structural variants where we need to be able to detect a pattern, like in a translocation that needs to align with the genome, it's a 50 kb minimum. The range of SVs that we can detect goes all the way from 500 base pairs to entire chromosomes. We detect aneuploidies, so chromosome arm deletions or duplications, entire chromosomes, and anything shorter than that can be detected with a copy number tool and also with our maps, again, as you can see here. Another question here, can this method detect mosaics? Yes, absolutely. We talked about the 1% allele fraction, so we can detect mosaic samples absolutely surely. Depending on the amount of coverage that you can get, we can go as low as 1%. And so, yes, we have plenty of examples of mosaic cases for FSHD, for a number of other genetic diseases. We don't have a lot of time left. We'll maybe answer two more questions. I am aware of advantages of these long-read sequencing platforms, but it comes with major limitations of high error rates, around 50% in case of PacBio and Nanopore. How accurate is optical mapping reads as compared to Sanger or Illumina? What is the error rate you have observed in general? It's a bit of a complicated question, of course, because we're comparing sequencing methods where you read the A, C, Gs, and Ts with a method like ours that just calls the structural variants by looking at these changes in label patterns. So yes, for Nanopore sequencing, the error rate is about the range that you called. For PacBio, it's actually a lot lower. I think the main difference between our technology and sequencing-based methods is that we are the only technology that can call these structural variants genome-wide, unbiased, with this very high sensitivity. So it's not a fair comparison because we don't give you single base pairs. We can't show you these smallest variants. But when it comes to structural variants, there's just no comparison in our performance versus the long-read sequencing platforms. And there's actually quite a few neutral studies, like from the Human Genome SV Consortium, that prove that. I think that is all the time we have. Any questions that we were not able to address during the live periods will be addressed by Ben Clifford or me via the contact information that you provided when you registered. So with that, I'd like to thank Ben Clifford for his time today and all of you for joining. We want to thank LabRoots for organizing today's educational webcast as well. On the top left in the resources tab, you have access to our Saphyr platform brochure and our SV white paper. And please contact us at info@bionanogenomics.com with any questions that you may have. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you that you share this with your colleagues who may have missed today's event. There's a survey that will pop up on your screen right now. We appreciate your participation, and with that, thank you, and until next time, bye-bye.